so much for the lovely introduction. Like he said, my project is investigating the role of the nucleus accumbens in social attachment. So whether it's making new friends or growing closer to your siblings, social attachments hold a deep significance to our species. Many neuropsychiatric disorders, such as schizophrenia, autism, and depressive disorders, accompany social attachment breakdowns. The prevalence of social attachments were also demonstrated during the COVID-19 pandemic, when many of us were either isolated or quarantined away from our loved ones and experienced hopelessness, depression, and anxiety. So to explore these social attachments or the neural basis of these social attachments, we use prairie voles as our model organism. Prairie voles are an excellent model organism to study human social dynamics because unlike traditional rodent models, they parallel human behaviors such as lifelong pair bonding and biparental care towards their offsprings. So why exactly the nucleus accumbens? Well, the nucleus accumbens is also known as the reward-related region of the brain, and it controls functions such as simple motivation and action to sexual reward. And previous studies conducted on prairie voles suggested that the nucleus accumbens is a crucial region of the brain that contributes to social attachments. They discovered that the nucleus accumbens has a greater density of oxytocin receptors in monogamous voles compared to promiscuous voles, which can allude to the belief that the nucleus accumbens is a vital region of the brain when it comes to social attachment behaviors. So the question we're trying to answer in this project is, what role does the nucleus accumbens play in social attachment behavior? And our two objectives is to manipulate the nucleus accumbens neural activity through chemogenetic manipulation, which is also known as DREADS, and to test the feasibility of chemogenetic manipulations in the prairie vole through CFOS immunohistochemistry. So the first thing we did was implement the DREADS, and this is where we basically virally expressed either an excitatory or inhibitory chemogenetic receptor in the nucleus accumbens of male prairie voles, and then provided a behavioral stimulus and a chemogenetic receptor ligand called clozapine N oxide, and this either excited or inhibited the neurons. And then we can test for the neuronal activation by conducting immunohistochemistry for CFOS. And CFOS is an immediate early gene that codes for a transcription factor that mediates long-term changes in neuronal functioning. And finally, we can test whether the manipulation of the nucleus accumbens activity changes pair bonding behavior by analyzing the tissue through immunofluorescence microscopy, which allows us to see the high resolution distribution and localization of specific cellular manipulations. So what we have here is the imaging results from the 2x immunofluorescence microscopy images. And to the left here, we have a male prairie vole that was injected with a GQ DREAD, which is the excitatory DREAD, but was provided no CNO and no stimulus. So there's no neuronal manipulation. So what we expected to see was no CFOS. And the middle prairie vole, or the second male prairie vole, had no DREADS at all, but was provided a stimulus. And what we expect to see from here is CFOS expression, but no DREADS expression. And then finally, the last male prairie vole was injected with a with a GI DREAD, which is the inhibitory DREAD, and was provided a CNO and stimulus. And what we expect to see from here is, And what we expect to see from here is DREADS expression, but less CFOS expression. So to give more information about this and break this down a bit more, we have broken down the nucleus accumbens into the left and right side. And to the top left of this table, I have a key kind of explaining what exactly these colors mean. So for example, this red fluorescence you see here is the M-cherry channel, which is the expression of the GI or GQ DREAD, which is the inhibitory or excitatory DREADS. The blue fluorescence is the DAPI channel, which is where it binds in the DNA to show the cell nucleus. And then finally, the green fluorescence is the expression of CFOS. So the male prairie vole that was injected with the GQ DREAD but no stimulus, as you can see, there is a lot of red fluorescence here, which is the expression of the GQ DREAD, but there is no CFOS expression. The second male prairie vole that had no DREADS and stimulus was provided. We can see these little green freckles here, which is the CFOS expression, but there's no hint or sign of a red fluorescence because there was no DREADS. And finally, the last male prairie vole that was injected with the GI DREAD and was provided a stimulus. It's kind of hard to see, but there's a little bit of red fluorescence in the middle here, and that is the expression of the GI DREAD. And around it is the CFOS expression, these little green freckles. And what's most important here is that the CFOS expression is less prevalent in the red fluorescence area, which is what we were expecting to see. So in conclusion, our imaging results may suggest that GI inhibition suppressed neuronal activity in our prairie voles, indicating successful implementation of chemogenetics. In the future, we'd like to finish cell counting to produce more quantitative data and finish scoring behavior to determine whether the nucleus accumbens inhibition changes pair bonding behavior. And finally, for some acknowledgements, I'd like to thank all of the CORI SSRP members who have given me this opportunity to explore my interests in research. And also a huge thank you to my mentor, Dr. Kimberly Long, who was honestly like the best mentor I could have asked for my first research experience. And Nanditha Krishnan, who was also an undergraduate working with me at the lab, who helped me navigate the whole lab process as it was really new to me, and all the Manoli lab members, and also, of course, the organization that funded my research. Yeah.